Welcome to our webinar.
Sorry, I cut you off there.
Got the warning. I was just about to say, we are going to record today's webinar since we would like to post this for others to watch after. So I'm going to be sharing my screen most of the time, so you shouldn't have to worry about showing up on the screen.
Go ahead and launch a couple polls here in the beginning while we're waiting for people to come in.
But are some firms taking a percentage?
I'm going to mute a few people that have some background here. Oh, whatever, that's gone.
Great, yeah. So welcome to today's webinar. We are going to talk a lot about Pipsqueak Pro and what this can do for your image analysis. Pipsqueak is something that we've been developing for a long time, and it's gone through a number of different versions. So part of what we'll talk about today is just some of the differences between the versions that are out there. But this is great to see a lot of new faces. And so first question here is really just, how did you find us? How did you find today's webinar? How did you find Rewire? Because this is a great chance to learn a little bit more about what we're doing.
Thanks so much for so many people answering here. I have a couple other quick poll questions. So if you want to answer this one real quick, I'm going to move on to the next one. But this is really helpful for us to know how best we're reaching people.
And if you'd like to stay up to date on these types of events and new releases, new things, promotions that we're putting out, we do have a newsletter that you can sign up for from our website. Or, feel free to send Christy or I an email and we can pop you on there. I'll have my email address up here at the end, but I'm also going to pop it into the chat. So feel free to grab it from there.
Okay, I'm going to switch to the next poll here. So we'd love to hear a little bit more about what kind of analysis you're doing and what kind of analysis might interest you in the future. So if we could just fill this out real quick, would be really helpful. We had a really good attendance today. It's great. Good to see there are a couple of people here from the experimental biology conference that we met just recently at the beginning of April. Welcome. Good to see you again.
Yeah, show of hands. I think you can raise your hand, on your Zoom link. Who went to experimental biology? Was anyone else in attendance?
There were a couple people from the How They Heard About Us poll that mentioned.
Yeah. Who's planning to go to Cell Bio?
Cell Bio in December.
In December, yeah. So a few months out yet. No one?
We'll hopefully see some of you at SFN in November.
Yeah.
Excited it's in San Diego this year.
Yeah. And knock on wood, everything will still be open. And we'll have it in person. I noticed SFN's email the other day. I was like, we are definitely doing this in person. I was like, I don't know if I would bet that hard on it.
I'm going to be optimistic, John.
Yeah, no, it was great to be in person for EB.
Yeah, EB was great. Everyone seemed really happy to be out in a real conference again. Okay, one last poll question here. Let me pull that up. Curious what you're currently using to do your image analysis. And if you're already familiar with and have used Pipsqueak AI or Pro, that would be helpful to know too. We definitely have some people here that probably looks like maybe quite a few people here who haven't used either version of our software. So it'd be great to show it off today.
Well, good. I think one of the benefits of Pipsqueak, and we'll definitely go into this here, is this was really designed out of the workflow that we used to do by hand. What I was doing as a postdoc by hand. So, we're really kind of using the same idea, a lot of the same methods as we use in ImageJ or Fiji, just coming up with an automated layer and our computer vision, of course, to help with the detection. So a lot of this should probably be fairly familiar to you, and we'll be able to go into it in depth. So that was the last question, right, Christy?
Yeah.
Great.
Yeah, so I can end this whenever you're ready to get started. But
yeah, well, let's hop into it. You can see my screen, okay, right? Yeah, great. Good. Yeah, so again, welcome to everybody here. Pipsqueak Pro is our latest version of our image analysis software. And what I like to say is, basically this software is designed to help automate the manual image analysis that you've probably been doing in ImageJ. So, hopefully this is something that can reduce a lot of the tedious, time-consuming, frustrating type of analysis that you're doing right now. And it really did come out of the work that I was doing as a postdoc, and I'll show you that here in a second. So today we're just going to talk a little bit about where this came from, why you might want to use Pipsqueak, and then we'll go into some specific examples of how to do single label analysis, how to do multiplexed analysis, because that's one of the functionalities that we really focus on. And then at the end, we'll talk a little bit more about the AI and how to customize the AI to be specific for the types of images that you're looking at. This is a topic that we won't spend a whole lot of time on today, but we are already planning an additional webinar in early June where we'll go into this in more depth. So stay tuned for more on that. So yeah, Pipsqueak has been around since about 2016. And we've always kind of used the name Pipsqueak. This was actually an acronym that came out of some contrived version or some contrived string of words that had to do with perineural nets and the types of analysis that we were doing back then. So since then, we've kind of shortened it to just call it Pipsqueak, Pipsqueak Pro. And so you can see a lot of our publications. We actually have a couple of links on the website just to Google Scholar. But most of these links are actually, or most of these citations are citing our original 16 paper, which is a methods paper. So since then, we developed an RRID for the software. So you should start to see publications citing this going forward. If you're about to submit a publication, this would be a great thing to include in there. But yeah, it's really impressive to see how well this has been utilized or how many people are using Pipsqueak around the world. And I think part of that is because of what we can do with it. So the types of analyses that it was really designed to excel at were looking at fluorescence or immunohistochemical imaging, staining coming off of like a confocal, a bright field, or a fluorescence microscope. And in the workflows that we were doing, and as I'm showing you here, a publication that just went out on Friday, so this is just in review right now, the types of workflows that we were looking at was really kind of post behavior, where we're looking at a slice of tissue. Our stuff was often brain tissue, but this is the software is not tissue specific, it's tissue agnostic. And so, we were going in and staining different proteins, looking at how they interacted and, different brain regions. But that was always a really difficult thing to do by hands because, we would have to take the confocal image and go in and circle the cells. And so this was all about standardizing the process within the lab. Quite honestly, because I wanted to be able to give it to some of the, interns or undergrads that we had coming into the lab so that I could, make my day a little bit more efficient as a postdoc. But this is still widely used in a lot of the research that we're producing. This paper is in association with the postdoc lab that I was in. And so, kind of where it began was a much more simple method. Some of the very earliest versions of Pipsqueak were actually a macro in ImageJ. And so our 2016 paper details that a little bit. But basically what we found was, these sorts of approaches were actually improving the reproducibility of our data. It was reducing variability between raters. And that was largely because it standardized the approaches, but it also helped standardize the detection of our cells. And the first way that we tried to do that, and I imagine what you've probably tried to do in ImageJ, is to threshold the images and detect clusters of cells, kind of using a thresholding method. And so what we found was that would work. That would work in some instances, especially if the cells were very distinct. But if there was overlapping of cells, thresholding, or even like segmentation approaches really don't work very well. It also wouldn't work well with faint cells because those would fall below your threshold. And then, of course, it wouldn't work well if you had artifacts running through your images or things like that. So, this was a nice approach for us, but it really didn't prove that fruitful. And so we moved towards an object detection based computer vision approach. And The reason that we did that is because I think there are a lot of examples of morphologies that really don't fit well into segmentation approaches. So if you've used software like Cell Profiler or Wicca, I think there are really great tools to segment an image if you have great clarity, little background, but those approaches often fail when you encounter, cells like this that aren't easily defined, something maybe like a microglia. This is a perinormal net. So just not an easy boundary to the cell or a lot of overlapping. And so object detection does a really good job of putting ROIs around these objects, and it can look for the morphologies that are very specific for what you train it for. And that really helps us pull apart some of these difficult to distinguish cells like this guy here. In the case of these perineural nets, I think this is actually a gap junction between two neurons. And so this is not something that we would have been able to pick up through a segmentation approach. And so, this method has really come a long way since its early days. This is actually something that we're putting together into a publication right now, where we've gone in and really identified how accurate our AI is, these object detection approaches compared to human raters. And so, one of our first concerns was intra-rater reliability. So, how much a person is varying through their workday, let's say. And the way that we measured this was essentially to ask a number of reviewers to go through and analyze the same 5 images, but over 20 instances. And all we did was rotate the image a couple times. And so, when you do that, a person is seeing the exact same image, but it turns out that they were not very consistent in the number of cells that they counted. This is just circling the cells here. Of course, the AI was very consistent with it, which is a great testament to what computer vision can do. But this is a good example of why an individual might be wanting to use computer vision in their software, computer vision in their analysis. The other, I think, good piece of evidence for using computer vision is Interrater reliability. And so here, this was looking at how much variability there was between raters, looking at a number of different images. So here again, we used 20 images, all unique this time. And I'm cutting out a little bit of data here. Basically, we compared some different AI approaches, but the takeaway that I want to show you is that our AI was very accurate compared to the human raters. And so we're coming up with about the same average as the raters. And this is important because we want our AI to be producing an accurate reading, but it was also much more precise because we were seeing, this significant decrease in variability, at least compared to our human raters. And so, again, this is probably more where you are wanting to deploy this type of software to other people in your lab, if you have undergrads or just even colleagues that are helping you out with your analysis. This is at least going to be producing the same analysis between each of the raters, and we'll talk a little bit more about that at the end, hopefully, and just in terms of how you can audit that type of analysis and see what people are doing. And so kind of the last thing that I want to touch on here before going into a little bit of a demo is just the different versions of Pipsqueak that you may have encountered, really just to address any confusion of what's out there. And so in its very earliest days, Pipsqueak was, an ImageJ, Fiji-based macro plugin. And this was, a nice way to go through the analysis, but it, I think if you've used it, you probably understand it was very frustrating because it would glitch out. Fiji was just limiting what we could do with an interface. And so about the past year, we developed our own interface that's really responsive. It's very intuitive, I think, but it's actually still based on the exact same approaches. So we're using some of the Fiji quantification library to actually quantify our images. And I think that's a reason why you can trust the type of quantification or analysis that you're getting, but hopefully not encounter the errors that you were in Fiji. And so you may see Pipsqueak AI or just Pipsqueak floating around in publications. We now call it Pipsqueak Pro. It's all the exact same analysis, just with a better interface. And so, if you're a longtime Pipsqueak user, I'd really encourage you to try this out. I think it's a fantastic way to get through your analysis even faster. And it's a lot nicer to use. just with any versioning of software, I'd probably encourage you to, wait to make that change until you're done with the project. But I think you have a better experience with it. And so, yeah, just to, kind of lay that out in a couple of bullet points, essentially everything that Pipsqueak AI can do, Pipsqueak Pro can do, but with a better look and feel, a lot more intuitive interface. And what you're going to start seeing is features that are going beyond what we were able to do in Fiji. So including some cloud synchronization and sharing capabilities, that are going to enable you to collaborate more efficiently with people in your lab or outside of your lab. Oh, and then I guess the one important point on that is that one license, each license you acquire are going to be able to work with any version of Pipsqueak. So, no concerns about having to switch over at the end of your license. If you have a Pipsqueak AI license right now, it will also work with Pro. And so you can head over to the website and download it. And so with that, I'm going to pop over and show you Pipsqueak Pro. So I'll give you a link here at the end where you can find a download and a place to get a trial license. But essentially, the very first time you download Pipsqueak, you're going to want to go through an installation and registration process that I'm not going to cover today because we did put together some YouTube tutorials. So if you're needing a little help going through that installation process, go check those out. What I will show you is we have a how-to link up here in our menu now, which will take you to this website that Christy put together. And it has a lot of information on how to get going, where you'll find your order number and things like that, some of those videos. So hopefully this will help out with that process. If it doesn't, feel free to reach out. We are here just for that. So the first time I log into Pipsqueak, I'm actually not going to have a whole lot of options other than to start a new project. What I have here now are a couple projects that I've already started on, but what I hope this reminds you of is maybe some of the image acquisition software that you're currently using on your microscope. We really designed Pipsqueak Pro to be project-based. So when you start a project, you'll be importing the images that you would like to analyze. You'll keep them together in an organized fashion and be able to go back and review those results. And so, here I might create a new project. I've already have, a couple here going, so I'm just going to open this one here. But if it was a new project, the first thing that I might want to do is either create some folders for organization. I can move images around in those folders, or I might want to just upload a whole directory as is. And this is, I have a choice to either choose individual pictures or a directory, but this is a feature that was really important to me as, kind of a power user, because when I export my images off of our, LASX software, off of our Leica microscope, I like to export them in directories because that keeps the different channels of my multiplexed image together, keeps them organized under, a single folder. And Pipsqueak is going to allow me to import those with that organization already intact, so I don't have to go back and sort anything. So this is a fairly small folder. I'm just going to choose this one. But what you see in here is two images. Each image has 3 channels plus a merged image. And so when I import this, I'm going to import both at the same time. So I'm actually going to choose, this maybe experiment folder that's kind of its parent, and say open, and then submit. And so this is going to start uploading them into this folder, and we'll see them pop up here in just a second. While we're waiting for those to pop up, and the reason that it does take just a second is because it's actually putting it into a directory for you on your Pipsqueak cloud. And so this is some of that functionality that you'll see come online here in the next couple of weeks. So here I've got some images that I've already imported. Oh, there's new ones. So here I can start to go in. This is one that I've already analyzed, but you can see, you've got the, grayscale image that's coming right off of a confocal. In this case, I've actually summed a Z-stack and imported it as a 2-dimensional image. Here are some of the other channels of that image, but, it's nice because I could go in and I can analyze individual stains, or I can analyze a multiplexed image. And so I'm gonna go back to just one other project here. And we can take a look at some of this analysis here, where we're actually looking at a number of perinormal nets in I think this is the infralimbic cortex, And so, you're seeing a lot of overlap, a lot of clustering of these cells that would be very difficult to do by hand. So in this case, I might want to go through and analyze all these images. All I have to do is go up and choose the type of AI that I want to point at it. And I have a couple of ways that I can do that. I can go into our new morphological selector and basically choose the type of thing that I'm looking for. And so here, we're really distinguishing our AI between large circular objects, small circular objects, in this case, non-circular objects. And so, this fits what I'm about to do. So I'm going to choose that one and I can press detect. And you'll see that's actually running detection here as we talk. And so in just a second, it's going to pop those results back up there. And great, this is looking pretty good. I also have the chance to go back and maybe try other models or other settings in here. And if this is your first time using Pipsqueak, I'd really encourage you just to take a few minutes and try out some different detection parameters to see what works for you. So, one thing that I might try to do is just to choose a different detection model. These are a little hard to decipher if you don't know, what that specific staining is, but I'll talk about that in a second. So I'm going to try a different detection model. I might also try changing some of these settings. And so my options here are that I can change how confident I want the AI to be in what it shows me. And so, essentially here, if I turn this all the way up, I'm going to see more ROIs pop up on my image. And if I turn this all the way down, I'm telling the AI that I want to only see the things that it is most certain of. And I think this is something that's important for the analyses that we're doing. I can also choose how much overlap I'm willing to accept. And so, a good reason that we may or may not want to see overlap is actually like this example here. And so I'm going to zoom in. Oops. I'm going to zoom in by holding control and then just scrolling in. And so what I can see here is, again, probably one of these gap junctions between two cells. And so I actually do want to detect both of those. And, for the type of analysis that we're doing on this image, I'm okay with a little bit of overlap in the ROIs, but I certainly wouldn't want to ignore that second cell there. So this is why I might want some overlap. But certainly there are cases where I don't. Like, I think this is actually just one cell. And so I'd probably just choose one of those ROIs. So I've made some changes. I'm going to press detect again. This is a nice little warning because now I've actually changed some of the ROIs. So Pipsqueak is aware of that and it's going to warn me before it just deletes everything that I did. So great. We're running it again. And this is going to pop up with some, I turned it way down. And so this is kind of a good example of, just seeing the couple cells that Pipsqueak was most certain of. So, that's, I think, the type of fine tuning that you can do to really come up with some detection parameters that work. And, what I like to say is the objective of this is to get 95% of your cells accurately detected with the AI. I think being good scientists, we never want to just completely rely on, what an instrument or what a piece of software is telling us. And so what I like to encourage all of, my colleagues to do and all of our Pipsqueak users to do is to go through these images, go through these results and double check what it's showing you. And so in this case, I think the AI pretty much got everything here. Again, that's that one cell that is maybe double labeled there. I don't think I want that. I also see a couple down here that I would like it to label. And so, I might add in those two ROIs there. But, that's, I think, the important, this is the responsible thing for us to do as researchers. And so now I like that. I think that's actually pretty good. And so all I have to do is approve my ROIs, and then I can export my results. And what that has done is actually popped these results right over into that folder where my images are saved. And so now I can see all of the ROIs that were detected. This is a spreadsheet that I opened up ahead of time, so I didn't have to make you watch Excel load. But same idea here, where I'm seeing 29 cells. I actually have an ID number for each one of those cells. And so this is kind of nice because if I go back over to my image, I can open up my ROI table down here. And now I can scroll through all of these ROIs that are present and actually see which one, was cell number 11. Maybe there's a reason that I don't think that one should be included. Maybe it's an artifact. And so now I can actually just unselect it. And that's going to remove it from our analysis if I export my results again. I've also got a number of metrics over here that I think are really important for the types of things that we're running stats on. I can see where my cell is, the XY central location of that. This is the center of mass of that cell. I can see how big it was, and I'll talk about how we can tweak that a little bit, or how we can set units for that here in a second. And I can see a general background that was measured for the cell. Again, here I'll go into that. What I liked to report, what I often publish, is actually the signal of all these ROIs. And so we define signal as mean minus background. So here I'm taking the mean intensity of my ROI, subtracting out the background, and that's telling me, these are just pixels above background. We also have a number of users who report integrated density. Here again, this is an intensity measurement, but it's actually just the mean minus background multiplied by the area of the cell. And so this is a great way to standardize the intensity that you're looking at across cells. So it's a little bit less dependent on how large the cell is. But hopefully from this Excel sheet, you should be able to move to stats pretty quickly, and start getting to the end of your project, because I think the last thing any of us want to do is spend a whole bunch of time, doing the image analysis as you're trying to get a paper out. So that's pretty much what we're seeing there. But let me point out a couple of those variables that I showed you. So the first thing that we can look at is the area of a cell. And so if you've used Pipsqueak AI, you might be familiar with how to set the area in Fiji. Now all you have to do is pop into our image analysis settings, and here I can determine what the area conversion is, in this case microns to pixels, let's say, and save it. And so what we'll see is that's actually going to change the area here. If I approve my ROIs, we'll see this area value change, because it's now it's now being multiplied by that pixel value. The other setting in here that I think is really important to understand is actually how Pipsqueak is subtracting out background. So again, if you've experienced Pipsqueak AI, then you're probably familiar with this grid of 22 boxes that we use to determine the background staining in an image. So what we're doing, our method is well published in that 2016 paper. And we actually have a great outline of that on our FAQ page on the website. But essentially what we're doing is we're measuring the pixel intensity of all these ROIs, and we're predicting that some of those ROIs are going to overlap with signal. So here's an example of that, where this box is right on top of a cell. So obviously that's not background. But we're looking at the whole curve of these intensities, and we're actually taking the middle third of that curve, assuming that some of the bright cells need to be removed, and actually some of the dark cells need to be removed. If I have rips in my tissue, or if some of these boxes fall outside of my staining, we actually want to make sure that we're excluding those. But now this is a chance where you can fine tune these. So let's say you want to move that box off of those cells, or there's maybe just a particular part of the image that you would like to measure background from. That's something that you can customize here. You can also go in, delete all of those, and come up with your own. I think one of the important things when measuring background is to, take a couple measurements from around the image. Don't do one, don't do, just 3 in one corner, but make sure that you're looking at the whole gradient of staining across your image. Once I've done that, determine what I want, all I have to do is save those background ROIs, and I go back to my detections. And voila, I'm still at the very end of my experiment. I can approve those again, export them, and we'll see those results show up here in our results file. So again, if you've used AI, this is probably a lot easier to go through, not so many clicks. Once you set some of these settings, what you should see is that you're done with your project and able to, or you're done with your image and able to go on to the next one.
John.
So, yeah.
There was a question in the chat asking about the existing biomarker detection models we have. Do you want to just show them the list and talk a little bit about the models real quick?
That's a great question.
Models.
Let's move over to a multiplex analysis, and we can start to look at that. I'm going to clear out some detections that I already have on here. But yeah, so our model library is really based on models that we've trained up in-house. So these are ones that we've vetted, ones that we think are very accurate. And so what you can see are, about 20 models in here that have all been published, that really range across a number of different morphologies. Now, some of these might not be familiar to you, particularly what parvalbumin looks like in neurons might not make a whole lot of sense, but the idea with this type of object detection is that it's really looking for a shape, a morphology. And so it's actually probably going to work across other similarly shaped cells. So one of the things that we're about to roll out in our next update or two is a little bit more explanation about what some of those models are. So here in our morphology detection models, you're actually seeing images of those. We'll be providing those for the rest of our models. So that should make it a little bit easier in the future to figure out what these are. In the meantime, I'd really just either Google what that type of staining looks like often, or give it a go and see if it works for your image, which is sometimes very surprising. What the AI will detect well. The other part of this is that these models that are deployed are ones that we've trained in-house. But one of the things that our users are able to do is actually to customize these models to be more specific for them. And so an example of what that looks like is we were just looking at those WFA images. And so I've created a custom model here just by pressing this button. What's nice about this is I can actually associate this model with a specific project. And so we're going to associate it with this PNN project. And when I do that, I'm only going to see images from that project. This is important because, when we train AI models, we really want them to only be trained on images of that class, biomarkers that are all of the same class. And so here I've pulled up all the images from that experiment that I've already analyzed. And I think this is a nice chance to double check what I'm putting into my AI model or how I'm customizing this AI model, primarily because AI is very good at reproducing what we teach it to do. And so if I teach it to do a bad analysis, it is always going to do a bad analysis. So this is a great example of an image that I would not want to include in my AI model because I've only annotated like maybe one of the cells in here. And so our AI is going to look at this whole image and decide that the rest of these things are cells that you don't want to detect. And so it won't detect them. I have another example over here that's a little bit better. I think when I made this demo, I didn't really detect all of these cells as accurately as I should have. So, that's something I would want to consider if I was actually using that. This is a good example of the types of annotations that should be going into it. But, anyway, the point of this here is that I can remove some of those images that I don't want to include. I can include the rest of them. And all I have to do is go down and press add. And that's going to pop all of these images over into my custom model. My custom model list has all the different models that I'm looking at, but and the one that I want to train up, I can actually see how close I am to having enough data in it for this type of object detection to train well. We like to kind of give a rule of thumb of around 2,000 cells being in your images. And so here, just with these couple images that I uploaded, I'm about halfway there. When I get up to that higher point, all I have to do is press train. And this is going to queue up a machine learning experiment on our servers that should return that result to you fairly quickly. This is a process that is becoming more and more automated all the time. And so our hope is that you can probably find a way to customize a model that's going to be very accurate for you using this really powerful machine learning technology. And so to go back to the question about the library here, just to make this a very long answer, is one thing that we are rolling out here in the future is the ability for you to share your custom models with the community of Pipsqueak users. Not only is this a great way to take some credit for the work that you're putting in, potentially even publish a method that you are producing in the lab, but also to let your collaborators do the same type of analysis that you are doing. So if you come up with your own custom parvalbumin model, you might want to make sure that all of your collaborators are using the same one. So look forward to that coming out. I think we're planning to have all of these features rolled out by SFN. So yeah, let's go back and take a look at this this multiplex or co-localization analysis, because again, this is the type of thing that I was really struggling with as a postdoc. One, because you'd have to circle 3 times as many cells or more, but also because trying to co-localize even across two images that are very dense can be really, really time consuming. And so, the method that I think a lot of people use for this is to flip back and forth between the images with all of your ROIs open in ImageJ. And if you've done that, I can tell you it's not a not a great experience. When you add in a third channel or a fourth channel or a fifth channel, I think that co-localization becomes impossible to do, by hand as a person. And so, here our software does this really well. And so what I'm going to do is I'm going to actually detect the cells in all three of these channels simultaneously. All I have to do is choose the model that I would like to apply to that image. So great, I've got those three. I also have my merged image here, and I'm gonna, basically just indicate that that's my merged image. And then all I have to do is press detect. And so, as I do that, this is actually detecting all of these cells at the same time. Voila, pops them all up. And basically, I'm already to the endpoint of my single label analysis. If I want to co-localize, I just press run co-localize, and it's reduced a couple hundred cells down into basically 7. Again, this is a chance for me to double check the results that are being shown to me here. We want to be publishing things that we know are correct. And this is a really cool example of why object detection works well on some of these cells. So here we're seeing, these two very tightly clustered cells pulled apart and actually co-localizing correctly using object detection. That's something that we probably wouldn't see with segmentation. So I've done that. I can export my results here again. And when I do that, these again are being saved over here in my my image folder. And so we can open those up and go through and see the the counts of cells, the intensity of those cells, and everything that I've shown you here. So this is Pipsqueak Pro in half an hour. There's a whole lot of functionality, a lot of very powerful features here. And what I'd really encourage you to do is to download our free demo and try it out. I think it's one of the best ways you can determine if this type of software is going to be useful for your analysis or not. And so you can find this a link to download a free trial on our website. I have the URL here, but also a QR code. We will publish this this webinar. So if you don't have a chance to get this now, thanks, Christy, popping it into the chat. Similarly, when you get going with this, if you need a little extra support, or if you'd like to do a personal demo with me, I'd be happy to run through some of your images. You can book a demo with me here, anywhere between 15 minutes and an hour. Seems to be enough time to start to look at how Pipsqueak can work for you. So yeah, maybe Christy, you can pop that one into the chat too when you've got it. Yeah, so great question, Emma, about pricing for students. We actually do have some student available pricing. This is something that we don't publish widely on our website, but I will tell you about it here because this is a secret just between us. Is if you are a student, we have a monthly license that we can make available to you for a slight discount. Hopefully this is a way for you to be able to access these tools if you need them and aren't able to get them through your lab. That pricing isn't available for labs or for industry, but reach out to us. There's actually a link on the Pipsqueak Pro page where you can contact us about that pricing. Cool. Yeah. Christy, could you pop this meetings?
Yeah. I'm working on it.
Okay, yeah. Yeah, so great question, Kathy, about licensing. One of the ways that we wanted to make Pipsqueak more accessible was to have it something that you can use on your timeline without having to buy a $20,000 license or something from Imaris. So our licenses are for a year long. We also have some monthly pricing available. Again, reach out to us if that's something that's interesting to you. But these are non-reoccurring. So if you're using a P card from your lab, from your department, it won't be charged again, but it will give you access to the software and all of the AI detection that you need for a year period. This type of analysis is really computationally heavy, and so basically that's giving you access to the cloud servers that are detecting your cells for you. You're able to train as many custom models as you would like, do as much analysis as you'd like. And what I'd like to really point out is, we are here to help you through any part of that process. And I think that's one of the things that differentiates Rewire from, some of those large image analysis companies out there, but also, some of the open software packages that are out there. I think it can be challenging to find the publisher of some ImageJ plugin sometimes. So we are here to help and make sure that you're successful with this analysis. We also have a lot of resources out there. I talked a little bit about the custom AI, the custom model training or AI customization. Christy's put together a really fantastic blog about this that you can find on our Medium blog channel. So here again is a link for that. I'd really take a look at this, kind of read about some of the best practices in training AI models like this, because that can really help with your success there. But we are planning to have another webinar just on training custom models sometime in early June. Keep your ear out for an announcement for that here in the next couple of weeks, let's say. But this is I think one of the things that you can do to really be successful in Pipsqueak is to just make sure that the AI is looking for exactly what you want it to look for. So in addition to this article, there's lots more on our blog. It's, I think, a really nice place to just kind of stay atop of what's happening in image analysis and in AI. Great. So yeah, good question here, Ella. In terms of providing annotated images, there are a couple different ways that we can basically teach this AI to be specific for what we're looking for. And so, let's go back to just looking at one of our images here. So this is the parvalbumin image, and essentially what the AI has shown me here, and this is we had pressed detect cells here using this parvalbumin model. And so what the AI is showing me here is exactly what it thinks is an accurate count of cells or detection of cells. But if I disagree with it, or if it's just not seeing what I want it to see, this is my opportunity to go in and change anything that I might want to change. And I might say that's not a cell. This actually reminds me, there's a great addition here that I haven't even pointed out, is that you can choose different shapes for your ROIs now in Pipsqueak Pro with more flexibility, some freehand tools coming out here shortly. But yeah, so I can make a number of changes to the detections in this image, and as I approve those now become the annotations that I'm training the AI with, and so that would feed back into the example that we looked at a minute ago with customizing the model. The other thing that you can do now in Pipsqueak Pro, this is something that you could sort of do in Fiji previously, is you can import your ROIs. And so what I'm going to do here is I'm going to import some ROIs that are not from this image. And so we'll just be able to see the difference here. So I'm going to go over, let's see, I'm going to find some of those really busy images that we were just looking at. And what you're going to notice here is I'm uploading a zip file that might be familiar to you if you've ever saved ROIs in ImageJ, because in fact, this is the exact same format, which provides 2 bits of flexibility. Either if you have, annotations, ROIs that you've already created from previous projects in ImageJ, you can import them right into Pipsqueak Pro. Or if you save your ROIs out of Pipsqueak Pro, you can open them up in ImageJ, you can open them back up in Pipsqueak Pro just by finding that zip file and saying open. In this case, I also have a couple choices. I can either add these ROIs to the ones that I'm seeing on my image in the background, or I can remove all of those ROIs and just bring in the new ones. So this is a nice way to audit maybe what a student is doing in the lab. You can open up their ROIs and actually see what they've detected, what they've measured, or to train a custom model. And so I'll say import here. And obviously these were not annotations from this image, but it kind of gives you an idea of how you can bring that functionality back into Pipsqueak Pro. And Christy, that's, I think, a really good point that there's a lot more detail to this in that blog article, and something that we can go into in more depth in June, or on a private call if you'd like to hop on a demo with me. So I will open this up to any questions that you have. I also want to, be conscious of everyone's time and how busy we all are. So if you can capture the 15 minutes of your afternoon, morning, and go on and do something else, feel free to hop off here. Like I said, Christy and I, and actually the whole Rewire team, are always available to help out. My email is here, also in the chat. That link to schedule a meeting is a HubSpot link, so feel free to use that and reach out to me, because I think one of the most rewarding parts of the software and building the software is seeing how other people can succeed with it and seeing what it can do for other people. So please feel free to reach out to us and let us help you be successful with it. So great. Yeah. Next question here. An experience with image analysis, or would it be challenging to get Pipsqueak to count and quantify areas of 2 different fluorescent colors? Yeah. So let me make sure that I'm getting your question right. So you're interested, oops, I'm changing the wrong screen. You're interested in maybe how we might look at an image like this and detect either 2 channels within this image, or look at just part of the image in 2 different analyses. And feel free to pop on with your microphone too, if that would help.
Yeah, hi.
Yeah.
So I would have images, an image with both red and green fluorescence, inside the same image, and I want to be counting the number of those areas, red or green, also, and also getting the area.
Yeah. Okay. So that absolutely is something that's possible to do. Really, the best approach to do that is to separate those channels out and look at them each independently. Kind of similar to what we were, ignoring this image now, to what we were doing, between a couple of these channels, because that essentially, brings them back into the merged image. And so we could actually measure the merged image if these were the pixel intensities that you were looking for. But to separate out those, 2 different stains is definitely going to be most accurate just by looking at each channel independently.
So would that need to be, could that be from a single image, separating them, those 2 out into 2 different channels? Or?
Okay. Yeah, we don't have the capability to pull those images apart in Pro right now. So yeah, you probably want to import those images in already separated out. But one thing I should mention here is we are working on the capability to actually have multiple classes in a single AI model. And so, maybe this kind of gets to your question in that, in this merged image here, I can actually see different types of morphologies, right? I can see like the big perineural nets, and then I can see the like parvalbumin or DAPI staining in it. And so we're actually working on the capability to have one model find both of those and separate the ROIs out for them. That's something that's not available yet, probably coming down the pipeline in the next couple of weeks or months. But that might be a way to get to I think the quantification that you're looking for. The other thing that we're rolling out into Pro here in fairly soon is the ability to just analyze part of your image. If you're an AI, if you're a Pipsqueak AI user, you might be familiar with cropping the image down. What we're going to enable you to do is basically pop in and say, I just want to look at this part of the image, or ignore everything outside of that. So this is something that you should see coming down the pipeline soon. We also have, let's see, I'm not sure if I can find my large example here, but we have the ability to look at tiled images and for our AI to detect the tiling within the image fairly well. So again, if you're a Pipsqueak AI user, this might be familiar to you. All we have to do is pop over into our image split functionality. And this is something that we're going to make a little bit more clear, but if you're an AI user, it should look familiar. Essentially giving you the option to chop this image up, kind of detile it, and have the AI look through each one of those little frames independently. This is a great way for the AI to, kind of take a really big picture image and find objects that it's used to seeing, sizes that it's used to seeing. So, this might be one of those things that you'd want a little tutorial on. If you're doing tiled images and want to analyze those tiled images, pop on a demo with me and we can kind of run through some of it and I'll show you how to make it work. But it is fantastic to be able to, see this large frame image and have all of your cells detected within it. It's pretty magical. So wonderful. Christy, did I miss questions there? I don't know where.
I don't think so.
Okay. Yeah.
I'm also giving our more general email for anyone. When you do try the trial, if you have any feedback, we'd love to hear it.
Yeah, thank you all for coming and joining us today. It's really great to see a lot of familiar names and some new faces. Yeah, reach out to us, let us know how we can help, and hopefully Pipsqueak will take some of the pain, the tears out of your image analysis and give you back some of your time. So thank you all for joining us, and hope to talk to you soon.
All right, thank you.
Great, and we can hang out here for just a couple more minutes in case any other questions come up. If anyone is hanging around. Grant, you want to update us on the timeline you were thinking for the sectioning, the image sectioning?
Yeah, so the subsectioning, or not subsectioning, the freehand drawing tools, we're almost done with those. We already have the support for polygon ROIs, so we just need to get the actual drawing tool working, and then we'll be able to actually crop out any region of the image that we want to. So functionality is almost there, so it'll probably be in an update or two.
Great. All good.
Anyone else have any more questions? No? All right.
Awesome.
Yeah. John, maybe we should end this then.
Yeah, great. All right. Well, wonderful to see you all. Thank you for stopping by, and Rewire team, I'll meet you over in Slack.
Sounds good.
All right.
Bye-bye.
Have a great day.
Good.